Welcome back, friends. Today we are going to continue with our part of enzymology. Today we are going to discuss regarding the enzyme inhibitors. Naenda kusoma kuhusu molecules zinazoitwa enzyme inhibitors. Kwa tutaanza kudiscuss introduction na kuona enzyme inhibitor ni kitu gani. And then after that tutaendelea na kudiscuss other aspects in part of enzyme inhibitors na kuona how inhibitor ni molecules wa aina gani. Kwanza tuone inhibitor ni kitu gani. Inhibitor kama ambavyo jina lina suggest, what they are doing is that they inhibit, wanasimamisha au kuzuia the enzyme catalyzed reaction to occur. So actually they don't prevent at all, but they decrease the rate of enzyme controlled reaction. Inhibitor, they decrease, wanapunguza rate of enzyme controlled reaction. So certain molecules can inhibit the activity of enzyme. Enzyme inhibition occurs in living organisms as the means of controlling enzymatic reactions. So kwa kawaida huwa inatokea kama ni njia ya kuweza kuzuia enzymatic reaction, ili kwamba hizi reactions zisitokee kwa spidi kubwa ambayo inaweza kaleta madhara kwenye mwili. Kwa sababu kila kitu kikizidi kinakuwa kibaya. Unaona? Ndio maana there is a rate which we need to maintain, which is suitable for the body to function well. So enzyme inhibitor, these are small molecules. Enzyme inhibitors are small molecules which can reduce the rate of an enzyme controlled reaction. So they are small molecules which can reduce the rate of enzyme controlled reaction. Many drugs and poisons also act as enzyme inhibitor. Dawa nyingi nishaambia kwamba enzyme ndiko ambako sumu pamoja na dawa zinaenda kufanya kazi kwenye enzymes. Unaona? So many drugs and poisons also act as enzyme inhibitors. Thus, enzyme can be poisoned by agents that form highly stable covalent bonds with groups inside the active site and thereby interfere with the formation of the enzyme substrate complex. Kwa inhibitor wanachokifanya ni kwamba wanakuwa wanaingiliana na formation ya enzyme substrate complex. Wanakuwa wanazuia enzyme substrate complex kufanyaje kutokea. Wanakuwa wanazuia enzyme substrate complex kutokea. Sasa tuone categories of inhibitors. Categories. We have two groups of inhibitor, namely competitive and non-competitive. Competitive and non-competitive. And the inhibition of either group of inhibitor can be reversible or irreversible. Kwa reversible na irreversible types of inhibition. Lakini inhibitors tuna competitive and non-competitive. Now let's see competitive inhibitors. What are the competitive inhibitors? Hizi kama jina linavyo suggest huwa zina compete, zinashindana. So these are the categories of inhibitor which compete with the substrate for the active site of the enzyme. Kwa hiyo zinashindana na substrate. Kwamba kama inhibitor watakuwa wengi, ndio watakaoingia kwenye active site. Kama substrate watakuwa wengi, ndio watakaoingia kwenye active site, kwa sababu wanashindana. So these are the inhibitors of a particular enzyme. The inhibitors have the shape which is sufficiently similar to that of the normal substrate to be able to fit into the active site. So this is the major feature of the competitive inhibitor. They have the shape which is similar to the shape of the true substrate. Normally it does not take part in a reaction, but while it remains there, it prevents the true substrate from entering the active site. So hapa kunakuwa kuna mfanano kati ya substrate na nani na inhibitor. Yule inhibitor anakuwa na shape inayofanana na shape ya substrate. Kwa kama shape ya inhibitor na shape ya substrate itafanana, manake inhibitor wakiwa wengi watabind kwenye active site ya substrate, na watabind kwenye active site ya enzyme. Wakibind kwenye active site ya enzyme, manake mwisho yote huyu enzyme atashindwa kufanya kazi. So the substrate and inhibitor therefore compete, wanashindana. They compete, substrate and inhibitor therefore compete for the position in the active site, and this form of inhibition is called competitive inhibition. So why it's called competitive? It's because the substrate and the inhibitor, they compete for the active site in the enzyme. That's why it is known as competitive. So thus, increase in concentration of one, either inhibitor or substrate, reduces the binding probability of the other. Increase in concentration of one reduces the binding probability of the other. Manake kama inhibitor ikiongezeka inapunguza uwezo wa substrate kubind, na kama substrate ikiongezeka inapunguza uwezo wa inhibitor kubind. So if the substrate concentration is increased, the rate of reaction increases. Unaona kwa nini? Kwa sababu substrate utakavyoongeza, manake itaongeza the probability of the substrate to bind in the enzyme. Now question, why should the rate of reaction increase under these conditions? Kwa nini rate ya reaction itaongezeka? Answer, increasing the substrate concentration increases the probability of substrate molecules fitting into the active site rather than the inhibitor molecules. Unaona, so to increase the substrate concentration itaongeza probability ya substrate kubind rather than inhibitors. Unaona, huu ndio mfano. We have the enzyme here, then this is the true substrate, this is the competitive inhibitor. So they both compete for the active site of the enzyme. They compete for the active site. Now, if this happens, that means this is the inhibitor, the enzyme, because the inhibitor binds there. And when it stays there, it prevents the true substrate from binding the enzyme. Like in this, the normal enzyme substrate complex, ambapo unaweza kuona hapo jinsi ambavyo ile substrate bind. That is competitive inhibitor. Remember, in competitive inhibition, what happens is that if we increase concentration of inhibitor, I mean if we increase concentration of substrate, the rate of enzyme controlled reaction now increase. Now let's see non-competitive inhibitor. Non-competitive inhibitors body. These are the ones which appear to react with a part of enzyme other than the active site. Kwanza zina react na sehemu ambayo sio active site. Since the non-competitive inhibitor complexes with a site other than the active site, it cannot be displaced from its binding site by competition of substrate molecules. Unaona, kwa hiyo hapa huwezi ukasema kwamba substrate zimekuwa nyingi, kwa substrate zikishakuwa nyingi basi zitamtoa inhibitor. No, in this case it's not possible. Unaona, inhibitor amebind kwingine kabisa, in very another place. Substrate pale amebind as usual, lakini uwepo wa inhibitor unazuia enzyme kufanya kazi. Kwa hiyo in this case ni kwamba inhibitor yeye anakuwa anazuia enzyme kufanya kazi. Hata kama substrate atabind, lakini hataweza kufanya kazi. So the substrate binds as usual, but the enzyme is inactive. Unaona, kwa hiyo tuna type mbili za inhibition kama tulivyosema. Tuna reversible na irreversible. Tuna reversible pamoja na irreversible inhibition. Reversible inhibition is the one in which an inhibitor binds to the active site of the enzyme with a weak bond such as a hydrogen bond which are easily broken. Kwa reversible manake ni kitu ambacho kinaweza kurudishwa nyuma. Ndio maana kwenye chemistry huwa tuna reversible reaction and irreversible reactions. Reversible, that means something which can go forward and backward. Reversible inhibition, that means ni ile type ya inhibition ambayo ni ile type ya inhibition ambayo hiyo type inafanyaje? Ina inhibit and bind with the enzyme with weak hydrogen bond which can be broken. So inhibitors affect the enzyme so long as they are attached to it. So they affect the enzyme so long as they are attached to it. For non-competitive reversible inhibitor which does not bind it to the active site, they do not affect the ability of the substrate to bind with the enzyme, but it makes it impossible for the catalyst to take place. As soon as they are detached, the enzyme can function normally again. You can observe this even what is drawn here in the diagram. This one tulisema kwamba unaona hapa kwamba huyu enzyme amebind substrate as usual, unaona, lakini the presence of the inhibitor disable the enzyme na inafanya enzyme kushindwa kufanya kazi. Presence ya nani? Inhibitor. Hii ndo tunaita a non-competitive reversible inhibition. Na ikiwa tu inhibitor ataondoka, basi enzyme inaendelea kufanya kazi kama kawaida. Akiondoka tu, enzyme inaendelea kufanya kazi kama kawaida. The rate of reaction decrease with increase in inhibitor concentration. When inhibitor saturation is reached, the rate of reaction will be almost nil. Kwa kama inhibitor saturation ikifikiwa, rate ya reaction itakuwa almost nil. Ni kama vile kutakuwa kuna reaction inatokea. Increase in substrate concentration does not affect the rate of reaction in this case. Na hiyo ndo feature ya non-competitive inhibition, kwamba increase in the substrate concentration high affect rate of chemical reaction. Example, cyanide is a non-competitive reversible inhibitor. It attaches itself to the enzyme cytochrome C oxidase, thereby inhibiting respiration. Hiyo ndo tuliyozungumzia cyanide kule kwenye denaturation. Ndio cyanide hiyo tunakuja kuizungumzia hapa kama inorganic chemical, inaweza cause denaturation, lakini pia inaweza act as inhibitor. Non-competitive irreversible inhibitor. Non-competitive irreversible inhibitor. These inhibitors combining permanently with an enzyme at the active site or as well. Either way, the change in the structure of the enzyme makes it ineffective as the catalyst. The change in the structure of the enzyme makes it ineffective as the catalyst. Unaona? Kwa hiyo ikiwa structure enzyme itabadilika, basi enzyme itakuwa ineffective na haitaweza kuwa tena kama catalyst. So the change may cause the protein or enzyme molecule to be precipitate, na inaweza ikawa precipitate, yani ikawa solid, na ikiwa solid maana yake haiwezi kufanya kazi tena. Mifano ya non-competitive irreversible inhibition. Kishakuta irreversible maana yake hiyo inaweza ikasababisha permanent damage to the enzyme. Mfano, ions of heavy metals like mercury, silver or arsenic, and certain iodine containing compounds in a very small concentration, they completely inhibit some enzymes. They combine permanently with the sulfhydryl group. Unaona? Haya, huo ni mfano wa kwanza. Mfano wa pili, kuna kitu kinaitwa nerve gas, DFP. Nerve gas, DFP. Hii ni diisopropyl fluorophosphate. Actually, this name is very long. Diisopropyl fluorophosphate. Diisopropyl fluorophosphate. DFP inaitwa nerve gas. Hii kitu kipuliziwa una dead hapo hapo. Inaenda ku inhibit acetylcholinesterase ambayo inavunja acetylcholine katika coordination tuna kuisoma. This is designed for use in warfare. Inatumika kwenye vita hiyo. Kipulizia maadui wakaipata hiyo, lazima wafe wote. It combined with the amino acid serine at the active site of the enzyme acetylcholinesterase. Huyu enzyme tutaenda kumsoma kwenye coordination kwa undani zaidi. Acetylcholinesterase. The enzyme breaks down neurotransmitter substance acetylcholine, and this will be discussed more in coordination. Sasa huyu ndio chemical mwenyewe anaitwa diisopropylfluorophosphate, DFP, the nerve gas. Inaua kabisa, inaua very quick hiyo nerve gas. So usitamani hata kuisikia hiyo nerve gas. Haya, from there now, let's discuss regarding allosteric enzymes and allosteric inhibition. Allosteric enzymes pamoja na allosteric inhibition. Allosteric enzyme, these are enzymes which are designed to change shape. The word allo means different and steric means shape. Allo means difference and steric means shape. So allosteric enzyme, these are enzymes which are designed to change shape. Hiyo ndio definition ya allosteric enzyme. These are enzymes which are designed to change shape. They are regulated by compounds which act as non-competitive inhibitors. They act as non-competitive inhibitors, or in other words nikwambie non-competitive reversible inhibitors, kwa sababu zenyewe inhibition yake ni reversible. Zikibind enzyme haifanyi kazi, zikiondoka enzyme inafanya kazi, so as to regulate the metabolic pathway in the cell. So normally the allosteric enzyme, they are used to regulate metabolic pathway in the cell. This compound regulating allosteric enzyme are called allosteric inhibitors. Zinaitwa allosteric inhibitor. Hizo ndo compound ambazo zina regulate the allosteric enzymes. They regulate the allosteric enzyme. So allosteric enzyme are type of enzyme which have two alternative forms of site. They are type of enzyme which have two alternative forms of site. One is active site with functional binding site. One is active site with functional binding site. Unaona? The other is allosteric site, which is inactive site, where the binding site of allosteric inhibitor alter the shape of the active site so as to become non-reactive. Kwa hiyo kuna part mbili. Kuna allosteric site pamoja na functional binding site. Unaona? So active site ambayo ni functional binding site. Kukiwa hakuna kitu kwenye allosteric site, hii inafanya kazi. Lakini kwenye allosteric site kukiwa kuna kitu, kukiwa kuna allosteric inhibitor, hiyo active site haifanyi kazi. So the allosteric inhibitor which acted non-competitive inhibitor reversible bind to the enzyme at specific site, yani allosteric site, which is well away from the active site. Unaona? The inhibitor modify the enzyme activity by causing reversible change in the structure of the enzyme active site. Kwa hiyo hapa structure ya active site ya enzyme inabadilika. Ni tofauti na kule kwenye non-competitive ambako active site ya enzyme ilikuwa inabaki vile vile. Hapa structure active site ya enzyme inabadilika. This in turn affect the ability of the subject to bind the enzyme. Unaona? Unlike non-competitive reversible inhibition, where the substrate binds to the active site, but the inhibition cause the impossibility of the catalyst to take place. Huku tunaona kabisa kwamba substrate haiwezi kubind. Sio ina bind hapo ifanye kazi, haiwezi kubind. An example of allosteric inhibition is found in the respiration process. The purpose of respiration is to produce ATP. When ATP is at high concentration, it inhibits one of the enzyme of glycolysis. Inazuia moja kati ya enzyme ya glycolysis. Allosterically, kwa ATP anakuwa nani? Allosteric inhibitor. ATP anakuwa allosteric inhibitor. However, when cell metabolism increases and more ATP is used up, the overall concentration of ATP decreases. Yani overall concentration of allosteric inhibitor decreases. And the pathway once again comes into operation because the inhibitor, which is ATP, has been removed. This is also an example of the end product inhibition, which we discuss here below. The end product of inhibition, which is used in the control of different metabolic pathway. Now look at this diagram. This is an allosteric enzyme having allosteric site and the active site. Now, if normal reaction occur, what happens? If normal reaction occur, that means the active site will bind there, and then the normal reaction occur in the absence of inhibitor. But in case if allosteric inhibitor bind at allosteric site, what happens? The shape or the active site of the enzyme changes, and then the substrate cannot bind the enzyme as usual. And if the substrate cannot bind the enzyme as usual, that means the enzyme become inactive until when the allosteric inhibitor is removed. That is the concept of allosteric enzyme and allosteric inhibitor and how they work. Now, one among the most common application of allosteric enzyme and allosteric inhibitor in the body is the control of metabolic pathway. Unaona, ni ile control metabolic pathway. Allosteric inhibition inatumia katika control different metabolic pathways in the body. Sometimes this is known as the end product inhibition or the negative feedback inhibition. End product inhibition or negative feedback inhibition. With many hundreds of reactions taking place in any single cell, it's clear that a very structured system of control of metabolic pathway is essential. Kwa hiyo there are a lot of chemical reactions which are taking place in the cell at the same time. So there must be a mechanism which will enable, must be a mechanism which will enable the control of these mechanisms so that they don't affect the cell. So if a cell were merely a soup of substrates, enzymes and products, yani ingekuwa ni soup ya substrate, enzyme pamoja na product, the chances of particular reactant meeting would be small and the metabolic processes ineffective. Unaona? Why so? Kwa sababu yani hiyo cell kungekuwa kuna mchanganyiko. Ndio maana kuna department kwenye cell, kuna mitochondria, kuna chloroplast, kuna peroxisome, kuna microtubules, kuna nini, and different organ and different reactions they occur. Why so? This is because different enzymes need different conditions such as particular pH, and it would be impossible to provide these conditions in such an unstructured soup. Kwa hiyo, for this reason, cell contain organelle. Ni kwa sababu hiyo cell inakuwa ina organelle, and the enzymes are often bound to the inner membranes in the precise order. So always the enzyme they are bound in the inner membranes of these organelles in the precise order. Unaona bwana? This increase the chances of them coming into contact with the appropriate substrate. So inaongeza chance ya hizi enzyme kuja kwenye contact na appropriate substrate and leads to efficiency. So the organelle may also have varying condition to suit specific enzyme they contain. Unaona? By controlling these conditions, an enzyme available, by controlling this condition and enzyme availability, the cell can control metabolic pathway within it. Kwa ni kwa njia hiyo, basi cell inaweza kufanya control metabolic pathway. When the end product of a metabolic pathway begin to accumulate, inavyoanza kusanyika, it may act as allosteric inhibitor of the enzyme, controlling the first step of the pathway. Inaweza ka act as allosteric inhibitor of the enzyme, controlling the first step of the pathway. Unaona? Thus, the product start to switch off its own production as it builds. Unaona? Product inaanza kuzima mechanism inayoiproduce product yenyewe. So the process if self-regulatory. The process is self-regulatory. The product is used up. Unaona? Its production is switched back on again since the concentration of product which acts as allosteric inhibitor decreases, and hence the enzyme become normal. Kwa hiyo ndio inaitwa end product inhibition. Presence of the end product in high concentration tend to inhibit the production of itself. This is called end product inhibition and is an example of negative feedback mechanism. Mfano P anaenda kumtengeneza Q, then R, then S, then T. Tuna enzyme 1, 2, 3, 4. Unaona? Sasa T wakizidi hawa T wakizidi wana uwezo wa kwenda kurudi kule. Sawa? T wakizidi wana uwezo wa kuja kurudi kuzuia yale pale. So tab come back. You see? Wanaenda kuzuia huyu hapa enzyme 1. Kwa T wakizuia enzyme 1 manake hizi zote zita stop. Hakuna kitakachotokea. Ila T wakipungua kwa kadri wanapozidi kutumika, end product as it is used up in the body, when they decrease, unaona, au T wakipungua kwenye mwili manake hii enzyme inakuwa switched on na maisha mengine yanaendelea. So explanation is our mchoro. Explanation: if T is produced in excess, it inhibits enzyme E1. Yule pale T enda kuzuia enzyme E1. This inhibits the production of Q and hence the whole metabolic pathway. Huyu akizuiwa maana yule atazuiwa na hence metabolic pathway yote. And hence the metabolic pathway stops. When the rate of the cell metabolism increases, if T is used up, unaona, as its concentration decrease, the enzyme E1 start to be active due to the decrease of allosteric inhibitor. Yani T ndio allosteric inhibitor. This results in the production of Q and hence the production of T. This is the mechanism of end product inhibition. Na hili ndio jibu la swali ambalo liko kwenye BS. Question 4.5 kwenye BS. In the coming session we will be discussing regarding the enzyme cofactor na types zake. Na ndipo tutakapokuwa tunamalizia kuhusu enzymes. Tukitoka hapo tutaenda kudiscuss water. Topic itakuwa iko over. Thank you and later wish you nice studies. Let's see you meet in the coming session.